घुमाना है हाँ, ताकि वो देख सके ना तो एक ऑन करो ऑन ऐसा ऑन अभिषेक तुम देख सकते हो क्या
Yeah, I can see
the whole this one lecture hall
yes
okay ऐसे छोड़ो साइड में बस बैठ जाओ सामने बैठो okay तो अरविंद को बोल स्टार्ट कर लीजिए
Yeah, we can start.
okay so I'm first of all I'm thankful to Abhishek who is rightly now he's placed in US and I think the time is 12:30 p.m. So he kindly agreed to give this talk and this is something I really appreciate although a lot of other commitment that he has. Okay, I will just give a little briefing about Abhishek's career. So Dr. Abhishek he is currently a MBL investigator and Can Zuckerberg imaging scientist at Marine Biological Labs, Woodford, Massachusetts. Okay. So Massachusetts and I mean Can Zuckerberg, this imaging scientist kind of things are very, very hard to get. So he's among the chosen ones, and I would just go a little bit beyond what he has done. He has done his master's in physics from Indian Institute of Technology, Kanpur in 2005, followed up by his PhD in physics at University of Massachusetts Lowell, 2013. And then he did his postdoc, a couple of postdoc, I would say, at Dr. Hari Sharof's lab as well as Daniel Lab. Daniel Lab is at Yale University, which is again a in a very big center for imaging and microscopy and biology. And National Institute of Health is something that we all know is a world leading institute, and he worked there with Harry. His research interests are basically in developmental biology, light sheet microscopy, imaging, optical microscopy. I would just go on to say, I mean, Abhishek is doing he's just kind of pushing the boundaries of science. I know him for quite some time, and he's building a new kinds of instrument, new kinds of optical imaging systems, microscope systems. And I'm hoping that some of these systems we are going to see in near future in the market as well. So that kind of research which has a lot of impact as I see. So Abhishek, I think thanks a lot again, and I would ask you to please go ahead with your exciting talk.
Yeah, thanks. Thanks, Professor Mandal. Thanks for inviting me, and certainly it's a great honor for me to join you all. You all can hear me in the back?
Just raise your hand so that he knows that you can hear.
Yes? Okay, good. So good morning. Sorry I can't be there in person, but still I'm glad that I have this opportunity to join you all virtually. And in next 20-30 minutes, I'm going to share some of the work we have been doing, and we are excited about that. I can't see my chat window, so please interrupt me if you have any questions or comments. Okay, so I'm going to talk about light sheet microscope and some of the applications, particularly what we have been working on. So I will touch upon a little bit development of light sheet microscopy, which I have been involved with in last, I would say 7-8 years, and applications, ongoing applications in my lab and some of my collaborators' lab. So I'm sure all of you have heard this word light sheet microscopy. It has certainly exploded in last 5-6 years. There is a good reason for that. because this has allowed us to do a lot of live imaging because it's gentle and fast. And that's why there are now almost 20 commercial systems we can buy, and probably more than 100 home-built systems, different variants of light sheet. Researchers, including Professor Mandal's lab, they have a few different light sheet systems they have developed for specific applications. So this is a field which has really exploded and continuously growing, both from technology development side as well as application point of view. And as you can see on my slide here, you are looking at butterfly ovary, which is one of the delicate samples I have imaged. And we have looked at this ovary to grow, and you can see these cells are doing some dance and they figure out their ideal position with time. And since it's very delicate and we want to watch this development, you have to be very gentle. And we want to watch this development for 30, 40 hours. So there are certain photo-induced bleaching and toxicity, and those concerns are there. So we have to be very gentle. The other example is a C star embryo, where we are looking at cell division at very early stage. And for this particular case, we are interested in tracking those nuclei and see after division how they position themselves. So for all sorts of these kind of applications, we need to get a 4-dimensional or rather 5-dimensional images. 3-dimensional space, 4th dimension is time, and 5th dimension is different colors, spectral information, so that we can learn whatever biology we are interested in. So why there are so many systems? Why we need so many systems? Because there is no universal system, and different biological questions will require different type of instrumentations and information. So like in case if you are interested in understanding single molecules or proteins, probably you want very high resolution information. But if somebody is interested in looking at corals, which is also very sensitive samples, but sample is big, probably same system what one can use for looking at single molecules of proteins may not work for that large sample, which will be few centimeter large. Again, there is a resolution, what type of resolution we need for particular biological questions. So all these things are dependent on what type of samples we are looking at and what questions we are asked. And based on that, you decide which system or which modality or which variant is suited for that particular sample and biology. And also you will learn, and probably you already know, different systems have a different sample mounting aspect. So that's important as well. If I have a good system, but if I can't mount my sample, I can't extract information I need. So that's also very critical aspect in light sheet, especially in light sheet microscopy, which sometimes we overlook or underestimate. The third aspect I will say is temporal resolution. Like if I am looking at, you are looking at in this particular movie, calcium dynamics. So if somebody is interested in looking at these calcium transients, you want to image few volumes per second or probably faster. While on the other hand, if I am looking at this developing C. elegans developing embryo, even I do 1 or 2 volumes per minute, that speed is acceptable or reasonable for looking at cell division and tracking those nuclei. So speed is also important, and speed, how fast we want to image, that depends on what type of biological questions we are asking. And last but not least is also signal and photo bleaching. In some cases, we have a lot of signal, a lot of photons. In many cases, we are starved of photons. So that also becomes a critical aspect. If you want to do long-term imaging, you have to minimize your photo bleaching. or you have to basically live with low photon, low signal images. And nowadays, I mean, it's good that we are doing this type of microscopy and imaging where we have a lot of computational tools available. So even with low or noisy image, we can apply some image enhancement or image augmentation approaches to further augment our image and do the analysis we need to do. But when we are planning live imaging or looking to image live samples, there are some of these constraints or some of these things we have to keep in mind. And that's why there is no one system which can work for all types of samples, wide range of biological problems, because any system is good or bad depending on what sort of biological questions we are asking and what type of sample we are working with. So why we need a light sheet system? I mean, we all have used or at least seen a confocal microscope. I mean, it's a workhorse microscope in most biomedical labs. Any lab you go, you will find a variant of confocal microscope. So what are the good things about confocal microscope? So confocal microscope gives you a good optical sectioning. And the reason it gives you good optical sectioning because there is a pinhole in front of detector. So it rejects out of focus light. It only allows photons from certain depth to reach to the detector. So that's a good thing. And in a way, confocal microscope is also a super resolution microscope. If you close the pinhole fully, then you will discard all your photons, but you can get a super resolved image. But what are the limitations? So point scanning confocal, the way you build any 2D image, you go point by point, as you can see in this schematic. So your speed is slow. And also, even you are collecting information from one point, your laser beam is going through the sample. So you are photo damaging and bleaching above and below of your point of interest from where you are collecting information from. So these are two limitations. It's slow, and phototoxicity or photo damage is more. And that's why we are looking for an alternative. If we want to do live imaging or fast imaging, what we want is speed and gentleness. So before I talk about light sheet, I just wanted to refresh your memory about lenses and detectors. So as you know, if you start using high NA lenses, numerical aperture governs resolution. So you can go high, highest right now you can go is roughly 1.7. It's an oil lens. But what happens is as you start increasing numerical aperture, working distance decreases. So like in this drawing, you can see on extreme left, that's a long working distance, but low NA lens. While on this right, it's a high NA lens, but very short working distance, which means this lens has to come very close to the surface or sample to collect photons. Even though you can collect a larger fraction of the solid angle, but it needs to come closer. All of you probably have heard or using a SCMOS or CMOS camera. In all our cell phones, it's a CMOS camera. So now the scientific SCMOS has really advanced and they give you a lot of good capabilities. So one capability particularly I wanted to highlight is these sensors, they have large number of pixels, 4 to 5 million pixels, but we don't need to keep all those pixels active or all these pixels in on state. Basically, you can select how many rows or how many pixels you want to keep active at any given time. So this flexibility or this capability gives you a way to basically read or collect information from certain region of that camera sensor. And this also we use in conjunction with the light sheet geometry to get confocality. And it's an electronic slit. There is no physical slit, but it's an electronic slit where you tell camera that I am only interested in collecting information from these many rows and reject all other information. So you are rejecting photons, but if it's required or if it's needed, basically you can get some sort of optical sectioning or reject out of focus or unwanted photons. So just keep these two concepts in mind, and we will come back when I discuss light sheet microscopes. So in my lab, we are interested in different types of tool development. And one aspect is light sheet microscope. So that's what I'm going to talk in next 10-15 minutes. But we are also interested in building and developing, making a confocal better or faster. And some other aspects in my lab, we are interested in super resolution imaging and turf microscopy, as well as some polarization and fluorescence anisotropy imaging. I'll be happy to talk or answer your questions related to these topics also. But today my focus will be discuss light sheet microscope with you. So I know some of you are at a pretty advanced stage, and probably you have used or built a system. So please forgive me because next few slides will be very introductory, and it might be redundant for some of you. But I just wanted to make sure that I refresh your memory before I go in more. We discuss more technical aspects. So how light sheet microscopes work? When you use a wide field microscope, you bring laser or illumination source, illuminate your entire sample, and you collect fluorescence from that sample. You don't have a choice to reject the photons. Whatever photons can make to the detector, they will be registered. They will be counted. So what happens in this case, because your illumination beam is going through the sample, you are exciting fluorescent proteins or dye molecules all over the place. And those fluorescent proteins are emitting photons, and your lens is collecting those photons. So you get, even though you have a limited depth of focus of your lens, but you also get a lot of out of focus light. And that degrades your image quality because you have a lot of unwanted photons superimposed on your area or photons you are interested in. Even though wide field can give you fast speed because you are looking at this entire plane or entire image you are collecting at once because you are using a detector, but there is no, in microscopy jargon, we call it, there is no optical sectioning because you are not slicing your sample optically to reject out of focus light. So light sheet is basically what you do is if you can shrink this illumination, then to begin with, you are only exciting part of your sample where you are interested in collecting information from. And this way there is no out of focus light or unwanted photons even to begin with. So how to shrink that? So what you do is basically you put another lens from this side and bring your illumination from this side and planar illumination. And that's why name is selective plane illumination or light sheet, because it's a sheet-like illumination. And then you have another lens which is in the orthogonal direction, and you collect fluorescence using that. And either you can move your sample to get a 3D, or optically you can move your illumination and your detection lens will follow. And this way you get a 3D information. So what you are doing is basically you are keeping the wide field detection part, which gives you a speed because you are taking this entire plane at once and you are mapping this on a camera. So you are getting a speed because of that. And you are limiting your illumination, excitation light. And this way you are getting optical section. And that's the reason why it's gentle. It's fast. Wide field is also fast. Light sheet is equally fast, but it's gentle because your illumination is limited, or it's basically confined in the region from where you are collecting information from. And that's why light sheet microscopes are both fast and gentle, and they are much more suited for a lot of live imaging. You can see in this drawing, when you are looking at wide field or confocal, you are bleaching your sample along the excitation path. While in case of light sheet, since your excitation is confined, you are bleaching very thin sliver of your sample. You are not bleaching entire sample. And again, now we are coming back because you need to put two lenses in this orthogonal geometry. So you need a long working distance lens. Otherwise, because of hysteric hindrance, you won't be able to fit two lenses. So that kind of poses some limitation on light sheet microscopes, because one of the requirement is to have a long working distance lenses. Hence, we need to sacrifice NA, because to have long working distance lenses, we need we can't have a high NA. So we are using slightly lower NA lenses than what you can use on a confocal um, on a light sheet systems. There are some examples. I may not have time to go. The people have developed single lens light sheet systems where you can utilize slightly higher NA, but still it's not like a confocal where you can go 1.49 or 1.4. It's still highest researchers are utilizing is around 1.2, 1.22 or something like that. This technique is old. I mean, first it was used in 1903. So we are reinventing the wheel, but we are reinventing in a better way because there are some other technological advancements. As I said, we have better lenses, we have better detectors, we have better computational capabilities, we have lasers. So put together all the other advancements, technological advancements are making the system more useful for biological applications. This slide, I just wanted to walk you through any light sheet system you can take, and you can dissect into three broader components. And what are those three components? Illumination optics. Basically, you need illumination source. Sorry, did I lose you guys? Can someone confirm if you can hear me? Jigme, can you hear?
Abhishek, we can hear. Please go ahead.
Okay, okay, good. Yeah, I thought probably we had lost connection for some time. Yeah.
Yeah.
Okay. Sorry about that. Yeah. So I was saying that any light sheet system you can take, you can divide in three broader groups. One is illumination optics, because we need lasers, we need a mechanism to create a sheet-like illumination. That could be a physical sheet, or even you can take a beam, scan it fast. Low NA beam, you can scan it fast and you can create a virtual sheet-like illumination. So we need those optical arrangements. You need a way to mount your samples, and there are different ways you can mount samples. Some people put samples in a capillary, like fly embryos or fish embryos. They are fine. A lot of embryos, they are fine in capillary. But if you are doing a cell biology, probably you want to do cell culture on a glass cover slip or those kind of things. So depending on sample, you need a sample mounting arrangement. And then you need a detection path where you have a detection lens. A lot of times we put a mirror for, it gives you some ease for alignment, then emission filter, tube lens, and then wide field detector. Commonly, SCMOS cameras are used with light sheet, but you can use any wide field detector which can give you fast frame grabbing speed. So any light sheet microscopes, basically you can dissect and uh, put in three building blocks. And I just said, because we need two for almost 90% light sheet microscopes, we need two lenses in orthogonal geometry. So that alignment sometimes is annoying, but also you don't have, you can't use high NA lenses because of steric hindrance. This slide is just to show you that there's so many different variants. People have probably used all kinds of combinations possible. And that's why there are more than 100 home-built systems and about 20 or 20 plus commercial light sheet systems. And each one has its own limitations and strengths. So it all depends what is needed. You pick or choose one geometry than the other. Also, there are some other optical engineering tricks you can play, and one is lattice light sheet, where you can illuminate your sample with a patterned light. And why you need to do that? If you do that and you can take multiple images, computationally you can fuse those images to recover slightly higher NA or a high resolution aspect of imaging. So basically you can combine structured illumination or a structured illumination like approach with a light sheet to get a high NA. You can also use multi-immersion lenses. There are a lot of neuroscientists, they work with brain samples, mouse brain or um, large samples, where they do the clearing to get rid of some lipids and all to make it optically clear. So you can combine this multi-immersion lens. These lenses are good in different refractive index media and image those samples. Basically, some of those clearing media, their refractive index is 1.54. So this type of lens is good in that refractive index. There are some clearing media where refractive index is 1.46. So you can use these lenses in that because you need to match the refractive index, otherwise you will have a lot of aberration. And again, there are another geometry where you can do multi-view simultaneously, like in this particular case from Philip Keller's group, where they illuminate from 4 directions and simultaneously collect information from 4 directions. That's advantageous, especially if you are looking at a sample which has a lot of scattering or absorbing centers or yoke. So if you're going from one direction, your excitation and detection, both those photons don't penetrate that deep. So if you can illuminate from multiple directions and capture from multiple directions and computationally fuse those images, you get a better perspective of that sample. So there are all types of possibilities in this case. I wanted to mention this particular system because when Hari started his lab, he wanted to do sample prep in a conventional way, the way we do for confocal microscopes or spinning disk microscope. So he came up with this idea that why not, we need to have two lenses in orthogonal geometry. It doesn't have to be like this. Why can't we rotate it and have in this geometry? And that's what he did. Basically, you can see there is one lens which is bringing illumination and the other lens which is detecting fluorescence, and they are in orthogonal geometry, and these two lenses are mounted on a motorized stage so that you can move them up and down for changing sample and all. And it gives you this orthogonal geometry for illumination and detection. So it's a light sheet system, but it facilitated mounting a sample in a conventional way. And that was also, that was very useful for cell biology and some of the samples which we can't mount in capillary and agros. So we wanted to make this system more versatile. So we developed a very compact fiber coupled, what we call is a dice beam dual view inverted selective plane illumination microscope. So basically you have two lenses and nothing prevents you choosing this lens for illumination and this for detection, but also you can switch the role. You can illuminate from the second and detect from the first. So basically you can get a two orthogonal perspective of your sample using the same optical arrangements. And that's what we did in this particular case. So basically you have two views, you collect two views, and once you fuse, you can retrieve better information than any one view. So the initial setup was done, which was pretty good for any physics lab, but it was not ideal if other labs, they wanted to adopt this technology. So as you can see in this left image, there are a lot of mirrors, galvos, and it has occupied a big optical table. But what we did then is we took this concept and miniaturized that. And the way we did it, basically, instead of using galvo and all, we used a MEMS mirror. So MEMS mirror, now you can buy these MEMS mirrors. They're two-axis mirror, and you can do tip tilt along this axis or this axis. So it gives you capability to steer your laser beam in this direction as well as in this direction. And they are very low mass. So you can really, as you can see in this scanner drawing, we really compacted it. And that allowed us to make our SPIM system footprint much more compact and easy to work with. So, I mean, there are a few other variants we have implemented, but in the interest of time, I'll just show you a few examples what we have done using such systems. And as I said, so this is the drawing where you can illuminate from lens A and detect from B. And if you do that, like you can look at the point spread function of fluorescent spheres. So as you all know, your lateral point spread function or lateral resolution is better than axial. Axial is always, axial in the sense along the optic axis, is always few times worse than lateral resolution. And we can't do much about that because that's inherent to physical optics, fundamental optics. So any confocal or any single lens system you use, if you look at the point spread function in XY, it will be better than Z. Z, it's always a little elongated. And that's what you're looking at here. If you look at view A, in XY, these spheres are nice and sharp, while in axial dimension, YZ, they are elongated. But what happens is the XY for A becomes YZ for B, because what is axial for A becomes lateral for B, and what is lateral for B becomes axial. And so basically, if you take two views and if you can fuse them computationally, you can get more isotropic. Basically, you can shrink your axial PSF using the information from the other view. So this is advantageous, especially if you are doing a cell tracking or if you are particle tracking. Having this more isotropic information in 3 dimensions helps you. Otherwise, what will happen is if two particles or two nuclei, they are next to each other along the optic axis, you may not be able to separate them or resolve them. But by using this two views approach or multi-view approach, you can basically separate them and find their centroid or find their track them in that respect. And I'll show you example in this particular case. It's in collaboration with my former colleague, Mark Moyle, where he's looking at the nerve ring formation in C. elegans. And the challenge for this type of imaging was because we were looking at in C. elegans embryo. These embryos, they start moving around a lot. It's a three-dimensional structure. So we wanted to get a nice perspective from all three dimensions so that we can see how this nerve ring is growing. It's a bundle of neurons. And the third thing is we wanted time because we wanted to do time lapse to see from the start to end when it starts growing and how it makes this connection. So we couldn't do this using confocal or confocal-like imaging. Light sheet, multi-view light sheet enabled us to visualize this nerve ring closing, and it gave us a perspective, three-dimensional perspective to understand what's happening, how it's closing. The other example, I'll give you this butterfly again. This is in collaboration with Nipam Patel and his lab people. So what they are interested in is basically they want to observe how these cells, initially, as you can notice, these cells are randomly oriented. But with time, they are doing this dance, going back and forth, oscillatory motions, and they find their position and they make a nice organized structure. So that's what they are interested in. And for that, they need to capture, basically, they need to get a time lapse so that they can connect all these dots. Otherwise, if you take a snapshot, you might miss about this oscillatory motion of these cells. So again, and this is, as I said, one of the most delicate sample I have imaged. So it was a very challenging to image for 30 hours without killing this ovary. And light sheet was very powerful in this case. It enabled us to look at that kind of sample for that long. I'll mention example of speed, why in some cases the speed is very important. And in this case, my good friend Evan and I, we were GRASS fellows, and we started this project. We said that when embryos hatch, they know how to crawl, they know how to find food, they can sense temperature. So we wanted to take a step back and say that this change, this circuitry or formation of this brain circuitry can't be radical. It's not that when embryo is hatching, all the synapses, all the neurons are getting wired. This must be taking place before hatching. So we started looking at basically calcium dynamics during the embryogenesis to understand what time these neurons start making connections, how embryos start behaving in a more coordinated manner instead of doing a random walk. So you can see this is an early stage embryo where we are looking at calcium spikes in muscles. When we looked at these same calcium spikes in late stage, just before hatching, that time their movement was much more coordinated. Then what we did is we looked at the span neuronal, basically GCAM is expressed in all neurons. And we noticed that some of these neurons are showing early activity, which means they are either forming a synapse or they are leading some of this circuit formation. So again, because of light sheet here, you can see we are doing few volumes per second for extended period of time. So systems like light sheet microscopes, which is fast and gentle, allowed us to capture that kind of data, which allowed us to dive in and understand the formation and of circuitry, brain circuitry in C. elegans during embryogenesis. Another example, this is all fixed samples, but these samples are large. If you have worked with cleared brain samples or organoids, or if you know your friends, they have worked, you know that it's a centimeter long. And if you do this kind of imaging on confocal microscope, it takes months. But on a light sheet, since you are going so fast, you can do in hours. So that's the advantage, even though it's a fixed sample, but we are taking advantage of speed in fixed samples, you'll say, I don't care. It's even it bleaches, who cares? It's a fix. But you can't wait for a month to look at data. But using light sheet, you can do this similar imaging you can do in hours. And that gives you, so there are certain advantages of having fast imaging speed in the context of neuroscience. And here I'm showing you two examples. One in collaboration with Kamran's lab, where they are interested in looking at all these neurons in a large mouse brain. And the other one is from Brian's lab, where they're looking at cleared organoids. And these organoids are about 4 or 5 millimeter big, but very dense. But because of light sheet, we can image it fast in a reasonable time. And we can also do multicolor. So, I mean, I showed you examples from embryos to neuroscience. Basically, there is a wide range of applications where these systems are playing important, or this technology is playing a very important role and allowing us to get the data we need for to our understanding. Now I will shift gear, and I wanted to highlight importance of interdisciplinary collaboration and how we can learn from computer scientists and make our systems or data analysis pipeline better. So it's very empowering. And here I'm giving you an example of a C. elegans embryo, which we have acquired on our light sheet data. On left, you can see it's as acquired data. And as you know, these SCMOS, there are some dead pixels. There are some pixels, they don't behave normally. So there are some bad pixels. Out of 4 million, there are about 1,000 pixels, they are bad. So if you take let's suppose if I have to do analysis, I'll be better off cleaning that data and then doing my analysis. Let's suppose I want to find these nuclei and track them. My segmentation and detection and tracking accuracy will be better on my augmented or clean data than raw data. Again, you have to be very careful. I'm not justifying or saying that we have to do this all the time, especially if you are doing any quantitative analysis where you are looking at measuring these signals and quantifying. You have to basically do it on raw data or as acquired data. You have to stay away from deconvolution or denoising, because as soon as you do any post-processing, you change all those photon numbers or counts and all. So if you are doing that kind of quantitative analysis, then stay away from these post-processing steps. But if you are doing, like in our case, we want to find these nuclei, we don't care whether they are 30 pixel or 33 pixels, that doesn't matter to us. What we want to know in this particular case is there a nuclei or not, and find the centroid. So we are better off cleaning the data little bit, because then our segmentation accuracy is higher. And you can see there are different steps in this case. You can take as acquired, you just correct for pixel, bad pixel. Then you can get rid of some autofluorescence, because a lot of times in fluorescence microscopy, you will see that there is a background or autofluorescence coming out from tissues or other parts of the sample. So those are not useful. So there are computational ways you can estimate and correct and clean for, account for that background. And that's what we are doing in this third panel, you can see. And then if you want, you can do further denoising or deconvolution to sharpen your images. So these are visually looking better. Again, if you have to do any quantitative analysis, stay away from that. But if you want to do segmentation or tracking or finding those blobs, then these approaches are, in fact, they are helping you, and you are better off using that. I'll give you another example. Oops. Sorry, it's not playing, but you can see this is as acquired images. And my collaborator, they are interested in these fine filaments, these actin filaments, but it has a big autofluorescence coming from yolk inside that. So what we did is we took few of these images and trained a model, and then computationally, basically, we segment that area which are not um, useful for us and account for that. And once you do that, then these images are much better, not only visually, but since our collaborator, they are interested in looking at these actin filaments, so they are not, these blobs are not blocking those filaments, so they can have a better visualization for their analysis. So these are, I mean, this is again light sheet data as acquired data, and then doing post-processing enhancement, which is allowing us to do our analysis, the analysis we are interested in little better. So combining your data acquisition imaging with some computational analysis or enhancement could be beneficial for certain applications. Not always, but could be beneficial for certain applications. Along the same line, I'll say that so it's my staff scientist Anthony is leading this project where we are interested in detecting cells. And as I said, I mean, we are interested in this embryogenesis. And one of the common problem which is common to all different types of embryo, where we want to know how many nuclei are there when they're dividing, where they are going. So basically finding those nuclei and tracking them is very useful for a lot of biological questions. So finding a nuclei, there are different flavors of that. In many cases, we just want to know where is the centroid. I don't care whether this nuclei is elongated or it's like a soccer ball or it's like American football. We don't care. I just want to know, is there a nuclei or not? And if there is, what is the centroid? While in some other cases, probably we want to know the shape. We want to know, is it elongated? Is it elliptical? Is it circular? Is it spherical? All these things. So again, depending on what information we want for our biological questions, there are different flavors of this blob detection or cell detection, you can call it, or segmentation. So we are developing tools basically to tackle such problems. And I'll give you one example where we had segmented this C. elegans nuclei in a semi-automated way. Basically, we do some segmentation, and if there is an error, we manually correct for that. But what Anthony is doing is Anthony is taking this raw data, cleaning it, and finding the centroid, and then he's generating this auxiliary image. Basically, each centroid is being represented by another sphere, a smaller sphere, so that all these spheres are well separated for tracking. So now what used to take us weeks, now we are doing in more automated fashion in a day. Still, we have a long way to go, because once latest stage embryos, when there are too many cells, they get so crowded and image quality is still not as good as we would like. And these are all light sheet data, but still image quality is not as good as we would like for doing this accurate segmentation and tracking. There are different approaches you can implement. You can do something called ensemble learning, where you look at, or you can ask computer to look at the same image again and again, 100 times or few hundred times. And then you can build a histogram. And this way you can minimize false counting, because at one instance, computer can say that, oh, there might be cell here. But if you do this 100 times or few hundred times, if it's false, then it won't count all the time. And this way you can reduce error and increase your accuracy. And thankfully, since we don't have to do, it's easy to ask computer to do these kind of things. You go home and let computer try the data again and again. Uh, as I said at the beginning, that typically we are taking 4-dimensional data, 3D. So this kind of segmentation you can do. How we are taking a 3D data? We are taking basically 2D slices and putting them together. So same thing you can do in segmentation. You can go slice by slice, and then you can merge them at the end. Or you can take the entire 3D data and do some axial deep blurring and deconvolution, and you can do 3-dimensional detection. So there are different ways one can approach such problems. The other thing why it's important to utilize computer, because in some cases it can do better than us. I mean, like in this particular case, probably I'll be confused whether there are 3 cells or 2 cells or 5 cells. But if we can train computer well, some of these patterns which we can't see, but computer can tease that apart and recognize that. So basically it can increase your not only efficiency, but also it can increase your accuracy. So there are different algorithms, different approaches. That's all. It's dependent on data. And in my opinion, in today's time, the computer science and algorithms have advanced so much, it's hard to imagine doing microscopy without sophisticated image analysis and image augmentation. So just keep in mind that you can make your microscopes better by taking advantage of other fields and other technologies which are progressing pretty fast. So I'm pretty much I have utilized my time. I'm running close, so I'll show few slides and I'll give you where we are going. There are so many developments being done. Is there any room for do further development? And I'll highlight this work we did when I was with Hari, where we put the sample on a reflective cover glass. So basically we have 2 lenses, but once you put on a mirror, you get a virtual images of both lenses. So like if I put a mirror in front of me, and if you look from this side, you can see my real image, but also you can see a virtual image. And so that's what we were doing. Basically, we were taking 2 real images and 2 virtual images. So we were getting 4 views using 2 lenses just by placing sample on a reflective, basically mirrored cover glass. And what you will see is with these 4 views, our collection efficiency was better because, as you know, fluorescence photons are emitted in all 4 pi. So some of those photons were reflecting back from that mirror and we were able to collect. And also it was giving us different views, different perspectives. So using 2 lenses, we are getting 4 different perspectives. And in this simulation, you can see that overall our collection efficiency was better, but also we were capturing a wider cone. So we were getting a little bit, sorry, excuse me, enhancement in our image quality. So where I want to go from there. So there is another field in material science, I mean, where people do all kinds of fabrications, including Professor Mandal's lab. So in material science, people know how to make this kind of trench or well, reflective well. Like this one, I have fabricated using KOH etching, where you on a silicon wafer, and this is a perfect mirror. So the idea is, can we utilize this kind of microstructure to do multi-view? So like in this particular case, we have placed embryo here. This is just a wide field image. and you can see one real image, but there are 4 virtual images. And since this mirror is at an angle, each virtual image is giving you a different perspective. So what we want to do is we want to combine this microwell fabrication with fluorescence light sheet imaging. And the idea is you can use a single lens, you can bring laser, reflect off from this mirror, so that gives you orthogonal illumination, and then collect multiple views simultaneously. So you are getting planar orthogonal illumination using the same lens, but also you are getting multi-view simultaneously. So that's the area. So basically, again, the bottom line, I mean, I'm giving you this example, but the bottom line is we need to borrow some concepts from other fields and see how we can use those concepts in a way which can make this modality better. That's the, I mean, whole purpose of showing this concept. The other area which I'm personally very fascinated about is orientation of these molecules. As you know, some of these fluorescent molecules, they behave as a dipole. And as a physicist, I'm always interested in knowing what is the orientation of this dipole in a three-dimensional environment. And what I learned is it's not easy to map for each molecule, each single molecule. If I want to map the orientation in a three-dimensional space, that's not straightforward. So what we have started is John Farukas, he proposed something called division of amplitude, where if you measure intensity in 4 different polarization orientations, basically you can compute, you can back calculate what is the theta and phi of that molecule in three-dimensional space. It has some degeneracies. I mean, it's not that you can get, there are some degeneracies, molecules. Let's suppose this is optic axis, molecule oriented this way will have a similar intensity distribution as well as molecule oriented this way. So there will be some degeneracies. So what we have started doing is basically we are trying to build an optical splitter so that we can image these 4 channels, 4 orientations simultaneously. And Anthony has also done some simulations to show, based on photon budget, what accuracy we can get, how accurately I can predict the three-dimensional orientation of these molecules. So we want to combine this with light sheet. The other area also related to polarization, I'm collaborating with my good friend Amit, and his expertise is making these meta surfaces. And they have designed a meta surface which can take an input beam and separate them in this Stokes parameter, what I described you. So basically, we also want that will make our optical layout and optical configuration much simpler, because you just need to insert this meta surface instead of doing this complicated optical splitter, and that meta surface will do all this splitting. So that's another area we are exploring. Can we combine this nanophotonics, where that field is very mature, and there are a lot of experts in that area. Can we combine that with light sheet to get information about the single molecule orientation? So there are a lot of, I mean, we have made a lot of progress, but still I feel that a long way to go to utilize the capabilities and potential of this modality for biomedical science or material science applications. So with that, I would like to thank my lab members, collaborators, and most important, all funding agencies, because without their support, we can't do. And thank you very much for your attention. I'll be happy to take questions if there is any. I can't hear you.
Can you hear me?
Yes, now I can hear you.
Yeah. So it was a great talk with a great
Oh,
clarity,
thanks.
and I think this is what all the researchers and students can appreciate here. I think you should consider writing book. It is yeah. I mean that helps a lot of lot in the field. Also, what we have witnessed today is the current research that is going on in the field. Okay, what is the recent advances that Dr. Abhishek could show all of us what is going on in his lab, and which is is a very great thing to have. I would like to add that very soon Dr. Abhishek will be a big star, and with all the kinds of waves that he is planning to make, and these are all some of these things I also even don't know. I mean, this is something that I think he's sharing for the first time. Although I know him for quite some time, but some of this I don't know. Overall, I believe that light sheet has the ability to explain. I think one day, how the light begins and how it develops with time. Okay, how it kind of builds up into a complete system. So, I will, Abhishek, can we take some questions?
Yes, please. Yeah.
Yeah. So I think we will take few questions if someone wants to ask. So if you have something that you want to talk about, you want to ask, you're free to ask.
And also you are happy to, you all are welcome to email me. I'm happy to set up a Zoom call and talk to you anytime. So please, yeah.
That's a great opportunity in that sense. So I guess we have a chat here. Someone, no, this is something else. Okay. So Abhishek, I do have to ask you. Have you tried have you tried?
Sorry, I can't hear you. You're.
During early development using light sheet.
Sorry, I can't hear you. Could you please repeat that question again?
I just wanted to ask, have you tried studying cell-to-cell communication during early development using light sheet?
Yeah, so that's a very good question. So we are looking at from two different perspectives. One is cell-cell interaction in monolayer cells also, because a lot of times cells come in contact. They're basically membrane will come in contact. They will traffic proteins and all, and then they migrate away. So we are looking at that also, but also in this embryogenesis, because some of these cells will go through asymmetric, what is known as asymmetric division. They will go this way, and then this daughter cell will divide this way. So we are trying to understand that as well, especially in fly embryos. Yeah.
Abhishek, just a continuation to that question. I mean, can we really figure out the morphology of cells using light sheet during active imaging?
Yes. In fact, if you look at the membrane, in fact, we have also looked at in cancerous cells, we have looked at the membrane ruffling and blebbing. Some cancerous cells, they bleb, and bleb in the sense they will extend their membrane in certain way in 3 dimension. And so we have looked at that. Basically, you get a fast 3-dimensional map of those cells. So if you label membrane, you can map that. Yeah.
Okay. One more question that I have is that I was thinking that you could mention a bit of plant biology that we can explore using light sheets.
Yes, well, that's very fascinating. In fact, there are a lot of papers you can see, in fact, including review articles, where people are using light sheet for plant biology, especially if you are looking at trafficking of minerals or actin filaments or cell migration. So I know one of my colleagues, he is utilizing a light sheet basically to look at plant roots. So there are a lot of papers out there.
Yeah. So yeah, do you people have anyone of you have any questions that you want to ask? Okay, Abhishek, I think we don't have any more questions.
Oh, wonderful.
And,
Thank you so much.
Really, yeah, it was really a great talk with a lot of clarity. So, if you share your email, I mean, I have it. So if you want, I can share with the candidates, and if
please
they want to
email,
really have a community.
please share my email, and I'm happy to answer your questions, or anytime, please feel free to shoot me an email anytime.
Yeah. Okay. Thanks a lot, Abhishek. I know. Thanks a lot for making the time, and let us thank the author also. Yeah.
No, it was a great honor. Thank you all, and enjoy your stay on that beautiful campus and a great lab. So
Yeah. Thanks
have
a
a great
lot.
day.
Thanks a lot, Abhishek. Yeah. Please. Thanks.